नमस्कार दोस्तों, वेलकम टू चैनल टॉक्सिकोलॉजी on board. Today in this video we'll be discussing another second part of this ICH S6R1 guideline, that is preclinical safety evaluation of biotechnology-derived pharmaceuticals. So we do not have questions on this particular guideline from the previous ABT recertification question papers, since these guideline-based questions are being recently asked since last couple of years, like last 5 years. So please comment whoever has appeared the exam in the year 2024, 25 or 23. If you come across any questions on ICH S6, S8 or S safety series or quality series of guidelines, we can discuss here. Okay, so I have taken these questions based on AI generated questions. Okay, so the question is we're discussing second part. So from the questions, we'll also try to discuss the important sections in the guideline. So the question is, which of the following studies may be omitted if scientifically justified? So if you look into this ICH S6 R1 guideline, no, biotechnology derived guideline, it is not similar to that of ICH S4 guideline or ICH M3 guideline, where you are not doing a checklist, where you are not performing all types of or battery of toxicological studies. Okay, we should not treat biologic as such like a small molecule or as a conventional molecule. Every biologic is different from another biologic, so because of which you have to do tailor specific or case by case approach. Okay, you're not doing any tick box. Okay, based on if that particular toxicology study is going to add any new information in the development of that particular biological product, the drug, then only you have to perform that particular toxicology study. Okay, that is the reason here in the case of biologics, no, reproductive toxicity can be, you can justify them. Even without doing performing these studies, you can justify which of the following studies may be omitted. You can avoid by if scientifically justified. But you should not just blindly avoid it. You have to give a proper rational, a proper justification. Why you are doing this study? Why you are not doing this study? And even if you do not do any this particular study, how whether it is going to compromise or how it is not going to impair the scientific or the product development. Okay. So right answer, first option is all toxicological studies. Option C is clinical studies. Option D is manufacturing validation. The right answer will be in the green text, that is reproductive toxicity. So I said S6R1 adopts a risk-based approach. It is not a checkbox, it is not a reproductive and developmental toxicity may be tailored. We have segment 1, segment 2, segment 3. So these type of products generally we do only EFD study. Segment 1 studies can be attached to the general toxicology study as an additional arm where you can check, not an additional arm, the same from the 3 month data only. You can look for male reproductive system and female reproductive system. From that, you can get an information. No need of doing a separate fertility segment one study. And coming to prenatal, postnatal also, um, since most of the time uh, we avoid what is it, this pregnancy, because these drugs, no, you have to think them similar to that of ICH-9 guideline. Okay, so if they are not used in this particular population, so no need of doing this particular type of study. I'm unable to explain about segmented justification. You only go back and check and comment in the section why we have avoiding prenatal postnatal segment 3, because I'm getting confused with ICH-9 guideline. I'm trying to give you the answer stating that the patient is in the later stage of last stages of survival. Please, that's not the right thing. I'll tell you that one. Coming back to so reproductive and developmental toxicities may be tailored, deferred, or omitted based on product characteristic, mechanism of action, nature of the target population, right? And clinical development plan, existing data. So if you see this ICID S6R1, no, it is flexible. Reproductive development studies can be omitted if scientifically they are justified, risk-based rationale support the decision. Okay, so this particular thing, no, here this section 4.6 they have given how you can avoid, how you can justify reproductive performance and developmental studies. So pause the video here and try to read it. Okay. Now we'll enter into second question. Second means the sixth, seventh question. This is genotoxicity studies. Genotoxicity studies is generally not routinely needed for proteins unless there is a specific concern. If you see these biologics, no, they are large molecules. Okay, so these large molecules going and intercalating with the DNA, it is very difficult. They cannot go and bind with the DNA. So that is a bigger challenge. So for that reason, That doesn't make any sense to do. Unlike like small molecules, where do a battery of genotoxicities like Ames assay, chromosome aberration assay, and micronucleus assay. Here there is no need. Okay, so rest of the options you read it yourself. Therapeutic proteins are large molecules. They do not interact directly with the DNA or enter the cell nucleus because of their large molecular structure. They cannot enter the cell nucleus and thereby binding with the DNA. Therefore, genotoxicity test generally not required for biotechnology-derived proteins unless there is a specific concern. What is that concern? Concern is if you have any structural alert, impurities, mechanism of action, and other risk factors. A risk-based approach is applied as per ICH S6 guideline. So this is the thing you have to remember. Okay. Now, for this, please, in the guideline, read section 4.7, genotoxicity. Here they have given this statement. It is not expected that these substances, large proteins or biologics, would interact directly with the DNA or other chromosomal material because it is unable to penetrate the nucleus only. So it will not be able to interact with this chromosomal material. Pause the video, read about section 4.7 in the guideline. Now we'll see question number 9, that is um, S6 addendum. So this I said S6 R X6 guideline, it came in, I think it came in 2000, I think 97, I guess. Second, this addendum was added in 2011. So together this guideline as well as addendum together now they are part of the single document. So this S6 addendum was incorporated into the parent guideline and it is renamed as S6 R1 Division 1. Okay. So guideline is S6 addendum S6 R1 2011. Okay. In 2011, the addendum was integrated with the original S6 guideline and the combined document together it is known as ICES S6R1. So this is the thing. So please read whenever you are reading the guideline, read along with this, read addendum also. Okay. The next we have another question related to kinetics. So, which pharmacokinetic parameter is particularly important when interpreting this toxicological studies for this biological products? The right answer will be, so generally whenever you are doing PK, we are trying to measure systemic exposure. So the right answer will be the green text, that is systemic exposure. So please try to read exposure data, whether these are the parameters like AUC, Cmax, they will help correlate observed toxicity with the drug levels and support dose selection for these clinical studies. Dose selection, so there are different methods for the biologics. Generally, we are going with MABEL approach. Okay, whereas for the small molecules, you are going with Noel approach. Okay, so under section 4.2.1, they have discussed about pharmacokinetics and toxicokinetics, how you have to do for biologics. Okay, then we'll go to next question, that is this question is related to carcinogenesis. So the question is the carcinogenicity studies for biotechnology-derived pharmaceuticals are. Yes, as I told you, this particular guideline is tailor specific based on case by case, whether this particular biologic will need any 2-year or transgenic mice assay. Is this going to add anything uh, in the product development? That you have to see. That's why it is case by case, tailor specific. So, right answer will be in the green text, that is, it is determined on a case by case. So, long-term carcinogenicity studies is not automatically required for biotechnology-derived pharmaceuticals. The need depends on the mechanism of action. We have to understand how this biologic, which particular receptor or epitope it is going and finding, what kind of triggering response it is causing, activation of which particular receptor it is causing. So the mechanism is important. Duration of the treatment. Is it once in a week or twice in a week, once in a month? And the biological activity. Therefore, the need for cancer studies is determined on a case-by-case approach. Okay, the factors which are influencing the need for cancer study will see mechanism of action and target specificity. Whether it is going to go and bind to certain epitope or antigen, that receptor complex, duration of the clinical treatment, short-term versus long-term treatment, biological activity like cell proliferation, immune modulation, findings from the repeat dose toxicity and other non-clinical studies. So this is also very, very important point. Instead of going for a 2-year mice or rat rodent assay, like that for the small molecules or transgenic mice assay, what we can do is that we can take a repeat dose toxicology study, 3 months. Typically, for the biologics, we are doing 3 month repeat dose toxicology study in the relevant species. So what you can see over there is that you can observe for pre-neoplastic lesions. If you see that there are some pre-neoplastic lesions, from that you will be able to explain whether this particular chemical have a potential concern for causing cancer. Okay, so like that you can eliminate or you can decide. Okay, case by case. Overall, the risk assessment using a weight of evidence approach. That's why you have to see which particular class is there. Is a similar class of drugs earlier has been done on which cancer study was done in that there is any positive findings are there. You can do in silico also, you can do direct laser like that. You can use the software and you can see, you have to see for the structural alerts also. You have to understand the mechanism. If it is not at all affecting a particular mechanism which is responsible for causing cancer, so like that you can give rationally, you can justify it for why you are not conducting this particular cancer study. Okay. According to ICH S6R1, preclinical safety evaluation of this biotechnology pharmaceuticals are not routinely required. The decision is based on a weight of evidence approach, considering the nature of the product and its clinical use. Okay, so here you can see under section 4.8, they have discussed in the guideline about cancer studies the importance of various factors like growth factors, clinical dosing, patient population, biological activity they have discussed. So you can pause the video and read about section 4.8. Let's quickly see why the other options are not correct. The option was always required. Option A, I said this S6R1 guideline, it does not require cancer studies for every biotechnology-derived product. As it will know, every biologic is different from that of the another biologic. Case by case, you have to see. Never required. Again, some products may warrant cancer study depending on the biological properties and intended clinical use. And fourth option is required only for marketing. This also you have to see. There are certain studies, for example, in the case of genotoxicity study. So if for this biological product you have done a gene tox study like AMSSA and it has shown positive, in that case also you can still continue the drug development because there might be certain mechanism of the drug which might interfere with the DNA. So as such, particularly you can see in the case of anti-cancer drugs, they themselves will cause cytotoxicity. So that doesn't stop from the drug from the development. So like that. So what I'm trying to say is that particularly for the ICH S9 guideline, whenever reading an ICH S6 guideline, no, there is a little variation with respect to this animal trials and requirements also for conducting phase 1, 2, 3 trials is little bit different from that of the marketing one. Because whenever you're going for marketing, you're doing it for a larger population. Okay. For that reason also, there is a difference. And coming to reproductive toxicity also, no, there is a difference with respect to region also. Japan is different, Europe is different, the requirements US is also there. So try to remember, integrate and try to read it. So option 4 was required only for marketing, incorrect. There is no such requirement if needed. Cancer assessment is considered during non-clinical development before approval. Okay, so that is the thing. So we'll see another last question. Last question in continuation with carcinogenicity only. Instead of we are discussing 2 year rodent essay, then we are discussing transgenic essay, 6 months transgenic essay. The question is, a transgenic animal model may be used when it improves pharmacological relevance. So this is very important. In case of biotechnology or biologics, what is happening is that you have to select for the relevant species. But in that relevant, if you do not come across any species from the available like mice, rat, rabbit, dog, monkey, if you don't have any species which is having that particular epitope or antigen to which this biologic can go and bind. So in that case, what you have to do, you have to also look for homologous. That concept also you'll see here. So ISH6R1 supports the use of transgenic or other alternative models when they provide more meaningful pharmacological and safety data relevant to the mechanism of action of human disease condition. Use the model that provides the most scientifically relevant information, not necessarily the least expensive or the quickest model. Okay, so when transgenic models may be preferred when the model expresses the human target or pathway. Okay, the idea of creating this transgenic model only is that if they from the available list of species, that particular epitope or receptor is not at all available, then we are trying to create a model, the mice model, rat model, We are trying to create a model where this particular receptor is there. Okay, so the model expresses the human target or pathway, right? The next is when it better mimics the human disease or mechanism of action. So from this transgenic model, if we are able to understand in detail about the mechanism, then this particular model will surpass the existing animal model when it increases sensitivity to detect potential risk. Okay, you're doing in non-relevant species or relevant species, but the sensitivity of that particular species to that particular biologic is less. In such cases, also you can go for transgenic models when it provides more relevant data for the risk assessment, when it supports better translation to clinical outcomes. So whatever the model or species whichever you're using, no, it should be able to translate that information towards human. Okay, finally, because the drug is going to the human. Okay, transgenic animals or other alternative models may be used. They enhance the pharmacological safety relevance of studies for biotechnology-derived products. So very quickly we see you can read this particular section under they are given this here expression the human receptors and homologous proteins. So this is very important. Okay, and let's quickly go through. It is a cheaper than standard species. Cost alone you cannot do. Then it has a shorter lifespan. This lifespan related this was with ICH-9 guideline, not this one. And human studies are completed. Animal studies are conducted before human study are not based on the completion. So this was few questions we have discussed with respect to ICH S6 R1 guideline, it is biologics guideline. Also important key exam tips you remember. Question areas will be, they'll ask you about with respect to relevant species. Which are the following relevant species they'll ask? Single species toxic studies for biologics. So generally, for conventional molecules, two species you have to do, one rodent and one non-rodent. Whereas in the case of biologics, what you can do is that you can go for single species if properly justified and rationally given, stating that that particular receptor epitope is present only in the monkey or only in the dog. Like that, you can avoid, you can file to the regulatory agency, either FD or EMA, with single species data. Immunogenicity assessment, very important. If your biologics, if your biotechnology-derived product itself is going to trigger T cell receptors or it is going to immune response, then your whole interpretation of tox data will be changing. That's why you have to add immunogenicity assessment in your battery of repeat dose toxicology studies. Gene tox or carcinogenic exception, as I told you, because of the large molecular structure of this protein, biologics, it will can't go and bind with the DNA. So it will you can avoid gene tox study, that is battery of gene tox study, and cancer also case by case until unless it is going to add some, if there is any alert is there, mechanism is there which is needed, then at that time only you have to do cancer study. Monoclonal antibody specific consideration, you have to there are there are separate sections with respect to maps also in the guideline. Read it. Read about anti-drug conjugate, monoclonal antibody, integration of the safety pharmacology into tox studies. Unlike small molecules, no need of doing a separate safety pharmacology in the case of your CNS as well as respiratory system. What you can do is that whatever the repeat dose 3 months toxicology study you're doing, in that only, in between the study only, you can also check for these parameters. You can run your FOB, you can run your safety pharmacology, HERG assay, you can run. Okay. Then risk-based science-driven study design. Unlike small molecules where there is a rigid design, there is a list, there is a tick box, check box. But in the case of biology, it is not. Okay. Thank you so much. Be curious. All the best for your exam.